I want to address this topic of um, really, I've titled it here, as you see, linking EoE and food allergy, but maybe it would equally well be titled, EoE as a manifestation of food allergy, or our efforts to sort of try to understand food allergy presentations more broadly and the ways in which they're actually a spectrum of disease, and in the hope that this will be consistent with FOSS's mission, and it's the mission of my lab as well, to improve both the diagnostic and treatment space by better understanding the underlying immunology. So our lab really focuses on the adaptive immune response, and if you're a card-carrying immunologist, you probably could spend the next 20 minutes better. Chris Olson told me that I needed to direct this to a broad audience, and she actually said fourth grade level, but I know this group is going to be a lot more sophisticated than that. I'm not sure I was reading in fourth grade at all, but this would be if you went to an undergrad immunology course a few years ago and sat in lecture for a couple of the days that were actually devoted to talking about allergy, because it still often gets short shrift in the overall context of everything there is to learn about the immune system. But you'd be presented, with a schema, something like this, where the innate immune system, and I'll put in the dendritic cell here as the representative, but of course, especially in allergy, perhaps the inputs and integration that the innate immune system provides by sensing the environment. And we now know, the nervous system has a huge role in that, but also epithelial cells and other things. And a lot of FOSS's effort, which I think was really has great potential to unlock new insights, is focused there. But we're interested in what happens after that those signals are integrated along with the presence of an allergen to instruct T cells that then drive B cells to differentiate and secrete antibodies. And in the case of allergy, IgE antibodies in particular, because those are the ones that can then subsequently arm mast cells and other cells that actually, upon re-exposure to allergen, degranulate and cause lots of the symptoms that we think of most often when we think about food allergy. So anaphylaxis and the other consequences of degranulation of those cells. EoE, in contrast, generally presents to us in clinic with, the consequences of long-term esophageal inflammation and ultimately organ dysfunction. So these are patients who ultimately, in the most specific way, present with food getting stuck. Earlier in life, it may present as chronic vomiting or reflux. And if you look at the tissue under the microscope, you see this influx of these allergy-related cells called eosinophils, hence the name, eosinophilic inflammation of the esophagus or esophagitis. But this is by no means the only thing going on. There's a lot of changes happening at the tissue level that have a negative impact on the barrier function of the tissue as well. And it's that long-term consequence of inflammation that leads to dysfunction. And a lot of the research, and I'm putting in this really fantastic paper kind of as a stand-in representative of that research, has focused on the role of the epithelium and sort of the epithelial unit and the ways in which EoE leads to its dysfunction, but also the ways in which its dysfunction may contribute to disease pathology. And in fact, what this paper shows is that there are individuals who harbor polymorphisms or mutations in immune pathways, in particular TGF-beta, that by themselves intrinsically can mimic a lot of the features of EoE. And so, one of the questions for a lab like ours actually is, does it even, why are we even studying the adaptive immune response, those lymphocytes that are really good at being specific to a particular target? And our answer to that is that, clinically, for a long time now, and this is an example from more than 10 years ago, we've known that EoE presents as a manifestation of exposure to dietary antigens for many people. And this is actually data from adults. It's generally, regarded as and more accepted that that's a feature in pediatrics. But here's data actually from adults, undergoing an elimination diet where 6 major allergens are cut out, and you can see improvements in their dysphagia scores, drops in eosinophils for most. And of those that get a response to diet, and I think importantly in this context of studying the adaptive immune response, recurrence of disease when those allergens are reintroduced. And we know from clinic, that we can subset these individuals into some 30 to 40% who specifically and only have disease that's triggered by milk, or it could be wheat, or it could be egg, or it could be some combination. And we now understand, the textbook has been updated, and T cells and how they're instructed by dendritic cells, even within sort of the slice of allergic disease, can be further subsetted by their function. And so it's really this T follicular helper subset that we now know, thanks to work of Stephanie Eisenbrocht, who's been one of the webinar series speakers earlier, and others that are responsible for driving, this high affinity IgE that can lead to anaphylaxis. But it's other subsets of T cells that have been associated for a long time with chronic forms of allergy like EoE, but also asthma and atopic dermatitis. And these cells are potent players to drive things like mastocytosis and eosinophilia, and ultimately lead to increased intestinal permeability. And our contribution in this area came from studying these cells, and it's summarized here as sort of the prequel, and I'll show you some of the data. We found evidence that a subset of these pathogenic effector Th2 cells, by virtue of expressing this protein called GPR15, are well poised to be responsible for driving EoE. And I think that, just before I get into the data, I think that I'll try to convince you that that has possible implications for future therapy and diagnostics. So here's a little of the data. I'll slow down a little bit just, to Leslie, if I get too jargony, just rein me in. But this is data that was derived in close collaboration and partnership with Chris Love's lab at MIT, another FOSSI scientist. And Chris had co-developed a platform for getting a lot of information about individual cells and the genes that they're expressing, along with their T cell receptor, which I'll describe a little bit more in detail. And by using this technique and focusing on the T cells in the esophagus in the top panel and comparing them to what we see in the small intestine, we could see a number of T cell subsets. And the one I want to focus on really among those that express this molecule called CD4, which means they're good at responding to outside antigens and instead of things like viruses inside, is this pathogenic effector Th2 population that had already been described previously. And we could see them really nicely. They're highlighted here in the top left. Each one of these dots corresponds to a single cell. We had about 5,000 altogether. And you could see these were very closely, well, I'm not showing here, but they're very closely associated with active disease. So in remission, these largely go away. During active disease, they increase in number and presence in the tissue. We could also take advantage of the fact that we had blood from these same patients and look at, using the same platform, the same technique, the T cell subsets that are present there, and indeed identify the corollary in circulation to those cells that are present in the tissue and have this allergic effector appearance, or phenotype as the jargon term. And here we take advantage of the fact that biology does this amazing thing when it comes to the immune system, and specifically T cells and B cells, which are responsible for recognizing, the billions of things that you might encounter over the course of your life. And they do that by sort of shuffling gene segments and combining them to make an incredible, array of different specificity receptors. And in the case of T cells, we call these TCRs, or T cell receptors. And each cell, when it comes out of its development, expresses one of those. But once it, by chance, recognizes something, say, like milk allergen, it expands into a population. And we could use those TCRs as essentially like little markers or barcodes to ask, what are the cells that have the same specificity in peripheral blood to those that are present in tissue, and what sets them apart? And when Duncan Morgan, the first author of this paper, did that, he found this small subset of circulating clonotypes, is the jargon, members of this expanded population of T cells that all came from, and have the same specificity, and ask what genes are overexpressed in that population versus the rest of the T cells. And what he found was high expression of this protein called GPR15. So that was the first time that anyone had done that, and the first time that we found that GPR15 ligand is actually highly expressed in the esophagus. The data were out there in public data banks as well, and that using an antibody, we could see that's epithelial cells that express that ligand, and you can see scattered T cells in the tissue that express that receptor on a protein level as well. And in fact, we've gone on with unpublished data by one of the postdocs in our lab, Momita Bomek, that these cells actually can use GPR15 ligand, the target, to migrate up a gradient across a barrier, and they do so more actively when they're coming from patients with EoE than active EoE than when that patient has EoE in remission or is a healthy control. So the red line being the active EoE. So how does this tie back to food allergy more broadly? Well, I want to tell you very briefly a second story about how we follow T cells using the same methodology over the course of the still only approved therapy for peanut allergy, and that's called oral immunotherapy. I'm sure many in the audience are familiar with it. One of the known adverse effects or side effects of OIT, I'll call it for short, is in fact eosinophilic esophagitis. About 5%, perhaps more, patients that are treated with OIT will develop EoE, and consequently, currently at least, the dogma is that that's a contraindication. If we identify someone who develops EoE during that treatment, we stop that treatment, and for most of those patients, the EoE goes away completely. So strong kind of causal evidence of linking the two. So over the course of this OIT trial that was funded separately, we took cells from circulation, stimulated them with allergen, looked at the subset that express this molecule called CD154 that enriches for effector cells, or 137 that enriches for regulatory cells, and we follow those populations over time, looking at their phenotype. And within the allergy skewed or Th2 population, we can identify indeed the expected subset. So for example, here are those pathogenic Th2s. They're called here, apology for sort of the jargon shift. Here we call them Th2A, but it's that same population. You can see some of the hallmark genes over here in this heat map that define them. We see the TFH skewed population. You can also see a regulatory-like population. The TFHs, the ones that I told you before, drive IgE production, very gratifyingly correlate with the level of peanut-specific IgE from the same patients. So that's consistent with what we know about their mechanism, and also consistent with what we understand about the mechanism of these mucosal effector Th2 cells. There was no relationship to the IgE level. And yet, in response to treatment, the patients that successfully went from being sensitive at a low exposure to having resistance to higher exposure had suppression of that Th2A phenotype. There was no change in the TFH that we could see consistently in association with disease. There was a little bit of IL-10 induction for the aficionados in the audience, and that might be relevant to why these patients have higher levels of IgG4, but that didn't really correlate with treatment outcome. Furthermore, if we sort of flip the question around and ask what's associated with treatment failure, one of the things that emerged, in addition to other sort of gene signatures that I don't have time to talk about and that we're trying to understand more, was this GPR15 signature. So we think this is a striking, finding, especially in the context of what I just showed you about GPR15 in EoE, and I think it helps to perhaps lead us toward a greater unification of understanding the pathophysiology of food allergy as a spectrum of disease, in which we might think of there being at least these two axes, and no doubt many more. And you guys in this seminar series have heard, really important work by Sarita Patil, my colleague here at MGH, focusing on the antibody side. But on the T cell side, we can talk about, I think, this axis of TFH necessary for producing high affinity IgE, and this axis of these pathogenic effector Th2 cells, perhaps operating cooperatively or in succession over age and development with innate-like cells as well, because these cells have the capacity to really drive, mucosal or barrier, like in the gut, changes that lead to increased permeability and perhaps greater sensitivity to allergen in that way. And so when you have, high levels in both of these axes, you end up here with this, very sensitive, low threshold form of food allergy. But also, as a consequence of OIT, some people, may go down this axis further and actually develop EoE, even as the IgE axis is suppressed. And over the course of life, we think also this can be useful as a paradigm for future testing and evaluation. And, it may prove to be wrong, may prove to be right, but I think it gives us sort of the things that we need as scientists, hypotheses to specifically test. The other way in which I hope that this work is going to be helpful is in potentially developing new diagnostics for EoE. If anyone in the audience is affected or has a child that's affected, you'll understand very well how burdensome it can be to manage EoE, particularly if you're interested in trying to treat with diet elimination, because it may involve multiple rounds of empirical trials of diet restriction and then an endoscopy and usually several rounds of that in order to get at the diagnosis. In early days, in an early collaboration with Michael Birnbaum's lab at MIT, we think that maybe we can exploit the expression of GPR15 on this subset of effector T cells to screen with novel tools, such as those developed by Michael Birnbaum, in which these viruses are engineered to recognize only a specific T cell receptor and can be generated with a library of food proteins, and then used to identify the specific T cells that are responding, and someday then give a diagnostic readout, such as this patient has allergy to egg or wheat, and hopefully reduce or spare patients repeated rounds of endoscopies. So I'll end there and acknowledge FOSSI's support, and very grateful for that. It's been important in several steps in this research, as well as my collaborators and colleagues in the lab. Thanks.
Thank you, Wayne. Thanks for, I think I see some questions in there. I'm going to start off by asking you a question that I thought about, because you talked about that there could be both therapeutic and diagnostic effects from the work that you're doing. And obviously that's a really interesting finding. Do you imagine that these new diagnostics that you just walked us through for EoE might eliminate the need for biopsies in the future? Because that could be transformative.
Yeah, it's going to take time. I think I hope so, is I think the fairest answer. And I think it's more likely and probably more likely sooner for the most common allergenic triggers, such as milk. So we have been, again, together with Chris's lab, building our knowledge of the T cell receptors that are specific to milk. There's a couple ways you could imagine this being a diagnostic. One would be very personalized, where based on an initial biopsy, the T cells are sequenced and the receptors characterized, cataloged, and then in comparison to that information, you do rounds of stimulating peripheral blood cells and look for the overlap. That would still be relatively labor intensive and I think pushes the limit of what may ever actually be feasible, although all of these things are getting easier, and there's obviously strong interest in personalized medicine broadly, including diagnostics. The other possibility is that as we build our knowledge of the T cell receptors that are specific to different targets, and we already begin to see shared TCRs between unrelated individuals who all have milk allergy or another allergy, that opens up the possibility of being able to, implement this as a diagnostic, probably with somewhat limited sensitivity, but good specificity. In other words, we may miss people, but if we get a hit, it'll be convincing, that have, say milk allergy or peanut allergy or one of the more common things. And so, it's going to be an iterative process, but that is exactly, one way in which I think that this work is going to be potentially impactful.
And on the therapeutic side, do you think that is it the GPR15, that protein that would be potentially a target here, or would it be something
Yeah,
else?
GPR15 certainly has some attractive features. G-coupled protein receptors in general are very, druggable, as people say in pharma. Um, it's easy to develop small molecule, orally available drugs that can block these kinds of proteins. There have already been hits from screens, looking at that. I think we have to understand the biology in greater depth
Yeah.
to know whether or not this is going to be an attractive treatment. So, for example, GPR15's expression is by no means limited to these allergy cells, and so there are potential off-target effects to contend with. However, one of the advantages in EoE, and we already exploit this for some forms of therapy, for example, when we use corticosteroids, is that you could imagine an inhibitor being, given topically with limited availability, to get to the bloodstream, and that might be a way. So yeah, I think GPR15 is an attractive target, although it will need more both basic understanding as well as development. I think also, though, understanding this subset of cells and its relationship, to allergy broadly, not just EoE, but IgE-mediated food allergy, is really promising. There are a number of targets that are unique to them. They have very interesting and specific profiles of metabolizing a number of prostaglandin-derived molecules. They express pretty high levels of IL-9 receptor. That's, there are already IL-9 targeting biologics that were in development for asthma and kind of, went away for more, as far as I can perceive, commercial reasons than, they were looked at a little bit in asthma. So I think that understanding the cell population, whether it's GPR15 specifically or other aspects of their biology, has a lot of potential in my mind.
That's great. That's exciting. I'm going to ask a question from the attendees, from Emily. She wants to know, should those who have successfully completed OIT and have been at some maintenance level for their allergen or allergens, in this case, peanut and tree nut, for a long period, in this case, years, and should they have a prophylactic endoscopy to confirm that there's no EoE, even if they don't have any clinical symptoms?
Yeah, that's a great question. My opinion, if Emily were under my care, would probably be at this time, no. Although I would certainly encourage, a healthy amount of scrutinizing for symptoms. Sometimes it does appear that some patients are essentially asymptomatic with EoE. I think, however, that I have personally relatively low levels of concern that left untreated, that's going to lead to long-term consequences that don't become clinically evident and that are, damaging or grave. In other words, I think the most likely scenario in someone who has OIT-induced EoE that is clinically asymptomatic is that eventually they will develop dysphagia, and that, as far as I see from the literature, and this sort of comes more from, our understanding of chronic EoE when it's undiagnosed, or how closely even we follow EoE and how closely we control it. Unlike something like celiac disease, where we know that those patients, if they're left, with unchecked inflammation, can develop, higher risk for autoimmune issues or even some forms of cancer. That does not seem to be the case with EoE, or for that matter, other chronic allergic conditions. So I think currently my answer to that would be that no, I don't think you need to monitor. I certainly don't think it's crazy to consider it. I think that I would be monitoring in the sense that I would be screening such an individual for symptoms that might initially be kind of subtle, like, dysphagia sometimes presents in young kids as just, they're drinking a lot of water with their meals, or they're taking a long time at the table.
Mhm.
Or unless you ask them, you don't really get a history. It's,
Yeah.
it's tricky. It's tricky. You don't always want to go looking for things that aren't there either, but I would watch them carefully.
Do we have time for one more question? Are you good, Wayne? Yeah?
Yeah,
Okay,
I've got time.
okay. So, how do treatments like Dupixent or steroids or diet compare in the way they affect these allergic T cells?
It's a great question. It's something we're still learning. I mean, I think for Dupixent, we have evidence already, and especially if we broaden my answer to that question to other allergic disease, such as atopic dermatitis or um, chronic rhinitis and asthma, that it seems the effect of these cells. One of the principal effects that drives inflammation is their secretion of IL-13 and things that Dupixent blocks. But the cells themselves appear to remain persistent in the tissue. Um, Dietary therapy, in contrast, the cells really, for all intents and purposes, go away and become less activated. Um,
Yeah.
Dupixent seems to be blocking their effects, but at least in the relatively short term, it's not making them go away. Steroids, I think, similarly, although I have less confidence in how many we've really phenotyped carefully on that treatment modality.
Yeah, okay. Okay, one last question.
All right.
On the OIT slide, you showed the peanut trial, and
Mhm.
you were talking about these specific, I think they were called like they were these honing gut T cells,
Mhm.
or these honing T cells. Could these be a predictor for patients one day that maybe shouldn't do OIT if it's going to lead them to have EoE?
Yeah, it's a great question. Actually, Caitlin Burke, who's faculty, junior faculty in our group, is looking at this question of a little bit more broadly than just EoE, but does the expression of GPR15 on this subset potentially at baseline, predict adverse responses, specifically GI type adverse responses to OIT? And our hypothesis is that it may, and could be specifically associated with EoE, or could be more broadly to GI adverse events. Or we don't know yet. The experiment is running, but it certainly would follow from the story as we currently understand it, that might be the case.
Yeah, very cool. All right. Well, thank you so much
Great.
for joining us today. We're grateful to have you as one of our investigators at FOSSI. Obviously, the research you're doing is critical to helping move our mission forward. We need, FOSSI has single-handedly transformed how we diagnose, treat, prevent food allergies, and we're not stopping until we find the cure. There are scientists and advocacy groups all over the world that are watching our cutting edge work, and it's solely funded by generous donors. So we need all of you attending today to help us power our next breakthrough. And you can donate now at foodallergyscience.org. And until then, keep on imagining a world without food allergies, and FOSSI will keep building it. Thank you all for coming today. Thank you, Dr. Streffler.
My pleasure. Thanks. Take care, everyone.